Hello, champ. So, yesterday we discussed a little bit about alpha helix and beta peptide seed, and today we will discuss more about it. And I promised that we will solve some mathematical uh, question. Okay, and along with that, we will, if time permit, we will see the Atkinson experiment, protein folding, and few things more. Right. So I hope you remember that I told you that the primary structure is basically the amino acid sequence. So if you see the amino acid as like a word, right? Then the primary structure is basically a sentence. Okay. And then the secondary structure, this is a paragraph. Okay. And the tertiary structure, that's a chapter and multiple chapter from a book. Okay, and today I will add a little bit more. So yeah, the main responsible bond, okay, for primary structure, or like for only the sequence, or only for making a peptide, that is the peptide bond. Okay, peptide bond, it is mainly responsible. The peptide bond is, of course, its covalent bond, right? Then secondary structure. The secondary structure, that's hydrogen bond, that majorly responsible for tertiary structure. That along with that, a peptide bond or hydrogen bond, both are there. Okay, because it's not nothing like that. The tertiary structure is coming without primary or secondary structure, right? So peptide bond plus hydrogen bond plus disulfide bond, disulfide bond, and the quaternary structure, that is mainly due to the hydrophobic interaction between subunit between subunit. So primary structure is due to peptide bond, that is the covalent bond. Secondary structure is due to hydrogen bond. Tertiary structure due to disulfide bond, that's again covalent bond. But hydrogen bond and hydrophobic interaction, these are not covalent bond. This is insulin chain. Insulin has two chain, A chain and B chain. If you see in A chain, it has one intra-protomer disulfide bond, and the chain A and chain B, they are connected to two disulfide bond. So in insulin, three disulfide bonds are there. That disulfide bonds actually giving stability and also responsible for its activity as well, right? So yesterday we started little bit about alpha helix. Today we will see a lot of things about alpha helix. So the major purpose of forming helical structure, suppose when some amino acids are forming something helix, that means for accommodating, for providing accommodation of a lengthy amino acid lengthy amino acid stretch. Okay, so in alpha helix, few things you should remember, that between two, so this is helical structure, and the helix is wrapping up in an imaginary axis, right? So the helix can be right-handed, the helix can be left-handed. How you can see that? Just through your thumb rule, right? So suppose the turn is in that direction, okay, and if you do your left hand, it will tell you that okay, this is left-handed helix, and uh, maybe this is right-handed helix, something like that. That will tell you the turn in that direction, that is right-handed, left-handed. You can think about that. Okay, but the major concern is that between two turn, that there is 3.6 amino acid residue between two two turns, and in generally the distance between two amino acid, okay, it varies it up to 3.5 angstrom, generally 3.5 angstrom. But as in helical conformation, So it want to accommodate more amino acid within a short distance, because suppose if you see the membrane protein, maximum of the protein has helical conformation. So the membrane is very thin. So you need to accommodate multiple protein in a small space. So for that, suppose in 3.5 Armstrong is the distance between two amino acids in extended form. Extended form. But same thing in case of alpha helix, distance between two amino acid is 1.5 Armstrong. Remember 1.5 Armstrong. That means one turn. So that we will call it as a rise. Okay, that means rise per turn. Okay, so the rise per turn here is, that means the distance between that point and that point, that is 1.5 into 3.6, that is 5.4 Armstrong. Okay, and how the alpha helix is stabilized? Due to hydrogen bond. Alpha helix is stabilized due to hydrogen bond formed between CO of N and NH of N plus fourth residue. Okay, that means suppose if there is let's assume there is 8 amino acid. Okay, one sorry, okay, let's see, M, L, P, so not P, let's do it as G. Okay, G, S, K, and again T, and A, and L again. Now this is first amino acid. Okay, so NHCO, right? Here in that direction there is NH3 plus. In that direction there is COO minus. Okay, now this is the peptide. Now the CO, the CO of M will make a hydrogen bond will 1, 2, 1, 2, 3, 4. N plus 4. That means 1, N plus 1, N plus 2, N plus 3, N plus 4. With that, right? Now that will make hydrogen bond with that, right? And it will form hydrogen bond with that. Okay. Now it will form hydrogen bond with that. And here K, the NH of K is involved, the hydrogen bond with that. Now it, the CO of K will form hydrogen bond with the next, but here is no next. That means this hydrogen bond is not possible here, because to make hydrogen bond we need another more amino acid here, right? So how many hydrogen bond you are getting? That is 1, that is 2, that is 3, 4. Total 4 hydrogen bond, 4 H bond from 8 residue you are getting. So you can try 12 residue, you can try 16 residue, but I am telling you in general, suppose if n is number of hydrogen bond, if, then number of amino acid residue in the alpha helix is n plus 4. Suppose if I am telling you in a question, if a peptide contain 36 hydrogen bond in alpha helix condition, calculate the average molecular weight of the peptide or protein. Okay, the question here I am saying, this is question number one, that if a peptide contains 36 hydrogen bond in alpha helical condition, calculate the average molecular weight of the protein. Okay, now you have to solve it. What you have, the information you provided, you have here number of hydrogen bond, that is 36. Now number of amino acid, you can calculate 36 plus 4, that is 40. Now average molecular weight of 40 amino acid is 40 into 110 Dalton, right? That means 4400 Dalton, or you can write 4.4 kDa. Similarly, if the same question I am telling you that 36 hydrogen bond is there, now calculate length of the alpha helix. Okay, this is question number 2. Simple. Here you know the number of amino acid. Number of amino acid equals to 40. Now length of alpha helix. Length of alpha helix is equal to 40 into 1.5 Armstrong. That means 60 Armstrong. Right? These two questions are quite important, and you can form multiple questions from here. Now you should understand few amino acids are generally known as helix breaker. We have to identify. See, there is no such hard and fast rule that this amino acid will make hydrogen, helix, alpha helix, or this will not. It generally depends on the surrounding of the amino acid. So suppose alanine is there, but in generally the propensity of alanine to form alpha helix is more or highest. But even it also matters the surrounding amino acid. So suppose there are G, 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 P, P, something is there. At that condition, obviously, the propensity of alanine to make some alpha helix is very less, right? So it also depends on the surrounding environment, surrounding amino acid. In generally, for helix breaker, you can consider as proline. So case 1, proline. Why this is called helix breaker? Because here immuno group is there. So the immuno, due to the immuno group, okay, so it can't make proper hydrogen bond with CO of other amino acid. Second, proline has like every amino acid has two conformations, cis and trans. Generally, proline prefer cis conformation. So somehow if cis conformation is activated, or if it takes the cis conformation, it will give uh, some kind of strain, okay, to the helix. So cis conformation. That's a constant for proline. Okay, what is the constant for other amino acid? So suppose there are few constants for D, E, H, K, R. What's that? Suppose you have, if this amino acid present like after 2 to 3 amino acid, then it's not a issue. Suppose if something is like that, suppose alanine, leucine and serine, then E, and then alanine and valine, isoleucine and then R. At that condition, these are not problematic. But problematic is when continuous stretch of positive or negative charge amino acid, because electrostatic repulsion, electrostatic repulsion. Okay, Third thing is that though glycine is favorable, so glycine is flexible. So you can think, okay, so as glycine is flexible, I can use it anywhere. But glycine is quite more flexible. That's what you actually need. Okay, so if there is some constant stretch of glycine, suppose something G, G, S, G, G, S, G, something is there, it will never form a helix. Okay, rather than it prefer to present as some random coil or some loopy like of structure, something like that. Okay, but generally small hydrophobic amino acid like alanine, valine, leucine, you can also consider isoleucine, these are preferred for making helix, alpha helix. Fifth thing is that those amino acid which are bulky group such as tryptophan, tyrosine, phenylalanine, these are bulky group. Bulky group. Actually, these are not preferred bulky group. Okay, rather some small hydrophobic amino acid are preferred. Okay. Now, in alpha helix condition, so suppose this is the alpha helix condition. At that condition, uh, so suppose the bond is forming here like that. Okay, this is the backbone, and all the uh, hydrophobic things. Okay, all the hydrophobic things are towards the uh, axis. Okay, that means all the hydrophobic bulky groups are towards the axis so that they are not exposed to water, or they are not water accessible. Rather, the backbone is water accessible. Okay, that's all about alpha helix. Now, another kind of structure is there available, that is beta sheet or beta strand. Okay, so what is beta strand? This is extended form. That means here two things are there. One is anti-parallel and another is parallel. What is anti-parallel? Suppose this is N terminal, this is C terminal of one strand, this is N terminal, C terminal of other strand, C terminal, N terminal of other strand, again N, C. This is anti-parallel. What is parallel? Something like that. N, C, N, C, N, C, N, C. In generally, beta strand varies from 8 to 12 amino acid. That's quite sufficient. It's not that much lengthy. Okay. So for anti-parallel, so what's happening here? So suppose uh, there is some, of course, okay, let's do something. NH3 plus CH. Now this is suppose R, and this is CO. This is again NH, again C alpha, R2, okay, again H or C double minus. This is one strand, and that here is also another strand, NH2N, CH, R2, CO, N, uh, H, okay, and again CH, R3, something like that, and then C double O something. So here the hydrogen bond is formed between NH and CO. So this, in case of alpha helix, hydrogen bond is formed within the helix. Okay, but here the hydrogen bond are formed intrastrand, not like, sorry, interstrand, not intrastrand. Okay, so hydrogen bond between two strand is responsible for maintaining beta sheet. Okay, In case of anti-parallel, in case of parallel, you can see the hydrogen bond. Okay, here in case of parallel, the hydrogen bond angle, like there is some strain is there. It's not exactly 180 degree. But if you do it in case of parallel, you can see the hydrogen bond is like that, 180. And hydrogen bond always prefer simple 180 degree. Okay, so suppose there is something O dot A something, and better than, it's quite easy. That's why anti-parallel beta strand is quite stable than the parallel, because the hydrogen bond forming angle is 180 degree in case of anti-parallel beta strand. But here it's quite uh, something strainful here, you can see, right? Okay, now there is two things you should remember in case of anti-parallel. Distance between two amino acid C alpha is 7 angstrom. In case of parallel, it's 6.5 angstrom. Okay, this is the distance between two amino acid here. Okay, uh, it's all up to beta strand. Now there is one thing is called loop. Sorry, we'll discuss loop later, but turn is important. Turn. So turn. Let's imagine you have something like beta strand here, another beta strand here, again another beta strand here. But you have to connect these two anti-parallel beta strand with something. How you can connect them? This is called turn. Suppose there is some alpha helix is there. Okay, now you have to connect it with another alpha helix. How you can do that? You need one turn. That is actually the role of the turn. Okay, so turn can be multiple times alpha, beta, gamma. We are people are generally interested more in beta turn. Okay, in case of beta turn, what happen? Hydrogen bond between I and I plus 3 amino acid. Amino acid. Here, uh, 4 amino acids are involved. Okay, so suppose this is I plus 1, this is I plus 2, and this is I plus 3. Now, the hydrogen bond will be from here to here, from between I and I plus 3. This is the beta turn. In beta turn, mostly proline and glycine. Okay, their presence make the beta turn, okay, more stable. And to make a beta turn, actually you need that. Why proline and glycine? Because proline, uh, the cis form of proline make it quite comfortable in the term, and the glycine is quite flexible. That's why proline and glycine is generally preferred for beta turn. There are two kind, like type 1 beta turn, type 2 beta turn, depending on the hydrogen bond forming pattern. And for alpha turn, so between I and I plus 2, and gamma turn between I and I plus 4. The hydrogen bond is formed between I and I plus 4. Okay, but the most common is a beta turn. Now also in some cases some loops are also there. Okay, so suppose this beta strand, alpha helix, when you combine all this structure, okay, all these things to make a protein. Suppose now our ambition is to make a protein. Let's make a protein. Now this is alpha helix, this is alpha helix. Now something is random, something loopy structure. Now we get one beta strand, another beta strand, here one beta turn, and for something like that, then again some kind of loop, again something, some structure like that. Okay, if something here, this is alpha helix, this is some anti-parallel beta strand, beta sheet you can say, and that's some random loop. Okay, loop doesn't have a specific feature. Okay, Suppose some GS, GS, GGSG, something is there. It will form something, some loopy kind of structure. Okay, and then this is the turn. So in a single protein, all these things are there. It's not that all these things are discrete. Rather, all these things are in a single protein to make a protein more compact, more functional, more structured. Okay, and that's okay. And then we have some more question. Okay, this is the Ramachandran plot here, as we discussed yesterday. So here, the phi and psi, I will try to just give you one reminder about phi and psi. So this is C alpha and C O N H again C alpha. So the angle between N and C alpha, so what will be continued, right? Again C O, right? The angle between N and C alpha is known as phi. The angle between C alpha and C is psi, and C and N is omega. This is a peptide bond. The peptide bond is partial double bond. All these things we know. Now these are dihedral angles. Phi, psi, omega. These are dihedral angle. Between so the phi and psi, this angle generally phi and psi varies from 1 minus 180 to plus 180, but all this angle of phi and psi are not favorable. So only the favorable angle uh, you some protein need. Okay, so this is a typical Ramachandran plot for polyalanine. Okay, this is for polyalanine. Here all the white region, wherever you can see the white region, that are totally not allowed. So this is not sterically comfortable. Okay, and the deep blue things where you can fit anti-parallel, parallel, and right-handed alpha helix, left-handed alpha helix, all these things are favorable. The deep blue, this is favorable. The fade that blue, here it is moderately favorable. So you have to do a little bit adjustment between the side chain, and that one, the faint blue, this is least favorable. Here there is quite steady hindrance, but still some structure is possible, but the white zone is totally not allowed. It's not sterically allowed. Okay, so as we discussed yesterday, the alpha helix phi is minus 5747. Okay, so minus 57, then minus 47, that's the region of alpha helix. This is the right R, this is the left-handed alpha helix. Now beta conformation anti-parallel minus 139 plus 1 here, and then parallel minus 190 plus 130, that region. Okay, and here also you will find collagen helix, that one, that means the collagen minus 51 plus 153. For beta turn type 1 and type 2, these are also given the phi and the psi. Generally for proline, okay, so that one is indicating I plus 1 residue. So suppose I and I plus 3, they are making bond, right? But I1, I plus 1, I plus 2 are also there. So here this is the phi and psi for I plus 1 amino acid and I plus 2 amino acid. Generally, as proline is there, it prefer minus 60 degree. Okay, it's quite rigid. Minus 60, minus 30 for I plus 1, and for I plus 2, that is minus 90 and 0. And that's also for beta turn. Okay, great. Now, in a protein, you can actually estimate uh, the amount of alpha helix and beta strand, some unstructured condition through circular digorism. Okay, as we learned this in some spectroscopy technique, right? Spectroscopy. So that the CD will help you, will tell you, okay, that whether your protein of interest have alpha helix, beta sheet, or status of folding pattern, status of folding or unfolding these things. So this is the alpha helix here, the blue one, the red one is indicating the beta strand, beta conformation, and the black one is the unstructured. So if you see it carefully, you can see the alpha helix is showing a good peak at around maybe 195, the positive peak 195 nanometer, and at negative peak it's showing as almost around 222 nanometer. For beta conformation, it's also showing something here like 192 nanometer, and here in between 215 nanometer. And for random coil, the negative thing is showing only in near around 198. This is a negative value. So through that, you can suppose someone can give you that picture and they can tell you which of the following is alpha helix, which of the following is beta conformation or beta strand, which of the following is some random coil or unstructured. So what is the length of a polypeptide with 100 amino acid residue in a single continuous alpha helix? So that is the 100 into 1.5, right? 100 into 1.5, that means 150 Armstrong. That's quite easy. Now the alpha helix also shows some polarity, okay, because in every amino acid, okay, they will show some polarity. So that polarity will be sums up. So suppose there is some C and O, or there is some CO in H. So there is a huge difference between the electronegativity of this atom. That's why they will show some polarity, and that polarity will so suppose 10 amino acids are there, all the amino acid will show okay some polarity, and then the polarity vector you can add them, okay, and if you add them you can see at the amino terminus, okay, it's behaving as a positive terminal, and the carboxy terminal is behaving as a negative terminal. Okay, the whole helix is now polar because it is the summation of the polar region. Summation of polarity of all amino acid involved in helix. Okay, that is the polarity of, sorry, polarity of alpha helix. Okay, now we will discuss more about how you can eliminate the protein amino acid sequence. Okay, little bit about mass spec, and then Sanger method for determining the N terminus of protein and different cleavage pattern. Okay, we'll see it tomorrow. And along with that, I just want to mention another more thing I forget, that in case of beta strand, okay, or beta seed, the side chain are either above the plane or below the plane. Okay, suppose NC or CN, because that thing above the plane or below the plane, that will reduce steric class. Steric class. Okay, that's all for today. Tomorrow we will discuss more regarding the amino acid sequence, like how we can elucidate that, and then the protein folding part, and then we'll jump into enzyme kinetics.